My guest today is Tom Charlesworth. He's the Director of Market Strategy and Corporate Development at biomodal. Tom, welcome to CC Life Science.
Thanks for having me. Pleasure to be here.
So, just for context, tell us a bit about biomodal, what you guys are up to, and then we'll get into the details of what you guys are up to.
Sure. Yeah. Okay. I can start at a high level. So we're a sequencing technology company. We're based in Cambridge in the UK. We've got commercial operations in the US and across and in Europe as well. Relatively small. We're about 110 people, and the company was actually we are the oxymoron that is a long-lived startup. We were spun out of the University of Cambridge about in 2012, and the company's been focused throughout its sort of tenure on the sort of interface between genetics and epigenetics and what epigenetics can add to genetics, really, and with a specific focus on looking at 5-HMC as an epigenetic mark. And then, around a couple of years ago, we focused on what we call our Duet technology platform, which is a platform that gives you either the 5 or the 6 base genome. The 6 base genome being your canonical 4 bases, A, T, G, and C, but also giving you single base resolution accurate calls for the methyl cytosine and hydroxymethyl cytosine, so the two sort of modifications, biologically relevant modification state, modified cytosine bases. And yeah, that's what we've been focused on for the past 2 years. We released the 5 base technology, which is called Duet Plus Mod C, early last year, and then the 6 base technology was launched earlier this year.
Nice. So that's exactly what got my attention is, well, one, just as a geneticist interested in epigenetics, because clearly we're learning a lot more about, how those modifications affect gene expression. I'm curious about how the cell knows where that information initiates. That may or may not come up today, but yeah, so we're going to talk about 6 base sequencing and epigenetics. So I'm interested in a bunch of things, as I mentioned, how genes are regulated epigenetically, what the 6 base genome can tell us, where do those things matter, and as I said, where does that information come from to initiate those modifications and do what they do. So describe the output and what you're able to see in a 6 base genome. And I'll just let people know, I'll put a link in the show notes for this to the video that describes how this sequencing is done. But tell us about the output.
Sure. So yeah, I won't focus on the kind of nuts and bolts of the technology. I'll focus on why it's interesting and why you should care. Yeah, sure. So the 4 base genome, the sort of traditional genome, fairly static, and it was hailed as the next big greatest thing that would tell us everything we need to know about where we came from and why we are who we are and how cells differentiate and how we go from a single genome into all these different kind of states that manifest across development and across our body, and then obviously going into disease and so on. That didn't really pan out as we hoped it would, so there was obviously there's more to the genome than just those 4 canonical bases. For a while, it was thought that there was one modification state of cytosine, the kind of meaningful modified base that had a sort of regulatory impact on how genes were expressed and how the genome was organized, and that was 5-methyl cytosine. In around 2008, there were a couple of seminal papers, one from one of our SCB members, Anjana Rao, that demonstrated that in mammals, you also get this other base 5 hydroxymethyl cytosine. And up until that date, technologies weren't able to provide you with the ability to distinguish between those 2 marks. And over time, as some of those technologies emerged, it became clear that 5-HMC, while not as prevalent as 5-MC, has a very different and distinct biological role to 5-MC. So 5-MC is generally associated with kind of repressing gene expression, closing down chromatin. A sort of classic example is closing down one of your X chromosomes. That's all mediated through methylated cytosine. Whereas hydroxy there's sort of a picture emerging of it being associated with regions of opening chromatin, of regions of increased gene expression. So functionally very different. There's a whole kind of enzymatic architecture, if you like, that underpins all of that, with different enzymes catalyzing the deposition of one or the other, transitions of one to the other. And so because they have those very different effects, as a company, we hypothesized that they must be it must be useful and meaningful to differentiate between the two. But there's also a literature that demonstrates that, as well as MC and HMC having these different functional roles, there's also an interaction between them and the genetic background. So if you think about the sort of differentiation cascade, if you like, through development, through sort of embryogenesis and so on, you always start from the same genetic kind of baseline. And then it's almost like a sort of pre-programmed cascade of opening and closing and expression and repression of genes kind of flows down from there. So there's from that you can kind of take away that there's a kind of relationship between the kind of full-based background and MC and HMC. But then when you get into sort of, I guess, later life, you have your body can respond to its environment, and those responses can manifest in gene expression. And so you also get MC and HMC integrating some of those responses to the environment. So that there's this whole you have a kind of relatively static 4-base genome. You have relatively plastic epigenome manifesting in MC and HMC, and it's all about the kind of how do those things relate? How does the environment relate to your epigenome, which also relates to your genome, and how can we get as complete a picture of that as possible? That was kind of the I guess hypothesis of why we thought that providing the 6-base genome was important. I can go on to talk about how that's kind of panning out with some of our early customers if you want me to kind of go straight into it, or we can kind of pause there and take a breath and see what else you wanted to talk about.
Yeah, let's pause there. I do want to know like how that's panning out. But yeah, it is fascinating. First of all, I mean, to even think about, our static 4-base genome, and yet there are things during development that are going to happen at a certain time, and that, even to think about how that timing is programmed somehow in a cascade of events that will happen. And then on top of that, at some point being able to respond to the environment as things change and then influence expression in the genome after that. So just to add a little clarity, if I understand correctly, what your platform does, I mean, you still do Illumina or whatever sequencing. It's all in the preparation of the DNA to be able to say at these cytosines, these modifications took place.
Yeah, exactly. So the 6-base genome, it's provided in a way that's compatible with short-read sequencers. We focused on Illumina for obvious reasons. It's the dominant player in the space. You have a kind of pre-sequencing workflow. You have software, and the output of the pre-sequencing workflow, obviously the sequencing and the software, is to provide you with at single-base resolution at very high accuracy, the 4-base calls for your 4-base genome, and then kind of on top of that the individual base calls for whether there's an MC or an HMC present, so it kind of gives you 3 states, if you like, at cytosines. It gives you unmodified cytosine, it gives you modified cytosine, and it gives you methylated cytosine, and it gives you hydroxymethylated cytosine. So the sort of logic of the assay and the software can deconvolute from a 4-base FASTQ that comes off the sequence into a 6-base FASTQ, to give you the complete genetic and epigenetic information from your sample.
Yeah. And so before we go into what your customers are doing, I guess we should explain that part of that, I mean, it requires, or does it, looking at specific populations of cells, because presumably in different cells, those modifications are different?
Yeah. So, yeah, I mean, yes. So you get, again, there's a literature that shows that you can get tissue-specific information from where regions are unmodified, where they're methylated, where they're hydroxymethylated. People have built tissue deconvolution kind of classifiers based on all 3 subsets of those data. There are papers go back, sort of arguing back and forth about which one individually might be most performative in a kind of classification, cell deconvolution classification problem. We would hypothesize that actually having access to all 3 of those information states is probably going to give you the best chance of best deconvoluting between tissue types. So yeah, there's sort of tissue-specific fingerprints, if you like, of methylation and hydroxymethylation. I think going beyond tissues into sort of sub-tissue cell types is challenging. I would expect there to be even more granularity if when we start to kind of dig down even deeper into the different fingerprints of the different cell types within a tissue.
Yeah. And I guess going back, I was probably thinking more at the tissue level than at, the more granular cell type level. All right. So let's talk about how are your customers using it? What are they learning?
Sure. Yeah. So there's kind of, our customers fall into kind of, I guess, three broad buckets really. There are those who are interested in the sort of fundamental principles, if you like, of the interplay between genetics and MC and HMC, and what each of those, what MC and HMC specifically do functionally within the cell. So we have a customer, Emily Hodges, who's more of a sort of developmental biologist who's interested in what the functional role of MC and HMC. We've got a webinar that we had put out with her, and we're going to have more later in the year, where she's been looking at through that developmental cascade, but in a neuronal sort of model system, what are the contributions of MC and HMC to the dynamics of the genome? So there's a literature that demonstrates that you can use epigenetic information to make some predictions and inferences about what's happening in terms of chromatin accessibility, and we've released some data that shows that you can do the same thing, and actually you can do it slightly better using 6-base data than you can using the sort of conventional data, which would conflate those two marks. That's looking at things sort of across the genome, so averaging out the impact or the utility of those two marks across the genome, which is interesting and useful. But actually, there's that by averaging things out, you lose a load of detail. You lose where the contribution more from one or the other, and that's where the interesting stuff kind of happens. So what Emily's been able to do is take some data where she's been looking at how chromatin accessibility changes over the differentiation of neuronal stem cells. She's previously had, if you like, plus 5 mod-C data that she derived from bisulfite, whole genome bisulfite experiments, and also ATAC-me experiments. And with that, she wasn't really able to. Well, there were sort of gaps in the relationship between methylation levels and chromatin accessibility. But when she started to use the 6-base information to probe those same questions, those gaps started to make sense, because if by virtue of being able to distinguish between MC and HMC, she could see that actually you were starting to see changes of HMC that preceded or aligned well with earlier opening of chromatin that otherwise would be invisible if you weren't able to distinguish between those two marks. So using just like a mod-C readout, you can't tell that there are these early changes happening. Because you've got that 5-HMC, you can start to see that there are specific regions of the genome that are important to that developmental stage, where you have a sort of spike in HMC that correlates really nicely with the opening up of chromatin. And from that, and she's done work looking at sort of relating it to sort of transcription factor binding motifs, and you can start to build up a really rich picture of the dynamics of like, how do you go from a closed, unexpressed state? How do you have something that kind of penetrates that, if you like, and starts to open it up, and then kind of continues this cascade of opening up? So that's been really exciting to see, just from a kind of fundamental developmental biology perspective. We also have customers who are interested in the 6-base genome in an oncology setting, and specifically in looking at liquid biopsy, where, again, there's a kind of literature base emerging that multi-omic information is far more powerful than looking at single omics in liquid biopsy settings. So trying to either look at kind of earlier detection of disease, but also looking at treatment response. We've got a number of projects. There's Sarah Jane Dawson at the Peter Mac in Melbourne, and we've got some other early customers in Toronto and also in Vancouver who are looking at different kind of problems in that space. Where they're sort of starting to see some utility is that you can, because using the 6-base genome, you're able to get as complete information as is possible from a cell-free DNA fragment. And so you can use that 6-base information to try and tease apart which DNA molecules within what is a heterogeneous cell-free DNA sample are maybe cancer-derived or derived from other kind of components of like there might be blood-derived and so forth. So if you can try and partition that out, then what can you learn from those fragments in terms of the kind of underlying mechanism, the underlying biology of a tumor. And these are, early stage, and it's something that we've also been taking a look at internally. We've published some data on some classifiers that we've developed to distinguish between CRC and healthy. And we hypothesized that because you're able to distinguish MC and HMC, because you can start to see these kind of early events of opening chromatin, you can get more temporal granularity in the dynamics of the genome. You can probably also get a lot more insight into mechanisms and pathways. And this is getting ahead of the data, but I get excited about it. If you're able to do that,
that's exactly where I want to go. Is like, what are the mechanisms and pathways?
Exactly. If you can kind of partition things, you can say, well, this region is becoming, there's HMC, so it's opening up, it's becoming demethylated, its expression's increasing. If you can start to kind of chain together those observations, can you identify novel pathways that you could then not only use as biomarkers, but also use as kind of points of intervention, or to use to understand mechanisms that might be driving drug resistance, for example. So those are kind of some of the questions that are kind of, I guess, swirling, if you like, and that I'm very excited to see our customers explore. So that's thinking about oncology. And then the final segment is looking at neurology. Neurology is fascinating for loads of reasons. Firstly, we don't know very much, so there's like loads of stuff we need to work out. Thinking about the 6-base genome particularly, it's really interesting because compared to other tissues, there's tons of 5-HMC in the brain, and no one knows why. No one knows why because we haven't really been able to measure it before very well. There have been tools, but they haven't been great. And so again, we've got some early customers who are looking at things like Parkinson's and brain tumors and thinking about Alzheimer's. Kind of similar questions, if you like. Like, can we work out from using the 6-base genome maybe some fundamentals about what 5-HMC is doing from a functional genomics perspective in the brain and why the distribution of it is different? There's some cool questions you could try and answer there. Can you discover new biomarkers for disease stratification? We know that in a lot of these neurodevelopmental disorders, trials don't work because you can't get in early enough, or because they're actually heterogeneous and you need to kind of subtype. So can you start to find biomarkers for that? Might those biomarkers, maybe you discover them in brain? Can you translate them into cell-free? We've got a customer looking at CSF cell-free DNA, which is pretty cool. And again, can you start to also piece together and tease apart mechanisms and kind of start to link things together? So yeah, those are kind of the three kind of, I guess, buckets of questions that people are asking of the 6-base genome and applications that
Yeah, so I mean, I think from my perspective, I mean, they're all interesting. The neurobiology part, which I know nothing about, is the most interesting to me.
Yeah.
And the developmental aspects of those cascades, and I guess the things, and maybe we're not there yet, but understanding what the signals are that, like, how does the cell or decide that we're going to modify the DNA here to open up the chromosome? What is telling it that now is the time, or that now is the scenario, regardless of what time it is, that it's time to turn this thing on? Like, how does information from the environment get in there is really the question I want to know.
It's a great question. I mean, it's a great question. I kind of, if you think about, because when you were talking earlier, there's kind of two problems, I think, when you think about that question specifically. You have the developmental biology kind of setting, where it's almost like you start at a baseline and then a cascade happens. And it's almost creating its own environment. And so that's easy. I think that's conceptually easier to kind of think about. But then when you get into later life and you have responses to the environment that often will manifest in disease. So again, one of our customers who's looking in neurology, she's interested in, this is Sonia Gandhi's group at the Crick, looking at Parkinson's disease. There's an interaction between aging and risk factors and how might that manifest in your epigenome. But Andy Feinberg's published papers on that you get kind of a increased stochasticity and entropy in epigenetics as you age. So is it just kind of almost like an element of randomness of the buildup of change? So life is you can think of the purpose of life as being controlling and minimizing entropy. Our bodies exist, and the bioenergetics of life exist to try and control chaos and to try and impose order. And as you age, that order breaks down and becomes more stochastic, and the entropy increases. So fundamentally is the purpose that we're trying to achieve in helping with disease, is it to restore the original order of our organisms. If you think about it,
fighting an uphill battle all the time, right?
Of course. Yeah. But if you think about it in those terms, then you can kind of, the effect of the environment can be either the effect within your body of just aging and kind of the accumulation of mistakes, but the accumulation of those mistakes can be accelerated by your environment. They can be accelerated by diet. They can be accelerated by, smoking, of course, accelerates mistakes essentially in your genome and your epigenome. And whatever else that you're doing, exercise can help to kind of almost reset the epigenome, I would expect, and so on and so forth. So I think that's at a high level how we could think about how the environment impacts your epigenome. It's either a general increase in randomness, or it might be a kind of more targeted increase in randomness because it's affecting particular pathways and that impact particular regions of the genome. So there are epigenetic editing companies. There are epigenetic therapeutics. There's one of our customers who spoke for us at the Festival of Genomics in London earlier this year. They're interested in epigenetic reprogramming, and what was interesting from their data was that they were looking at allele-specific methylation. So that's where you have within a sample you have two different genetic alleles, and you can have different methylation levels associated with those alleles. You can almost uniquely get that from our data because the genetic and epigenetic information are on the same fragment of DNA. They're on the same read. So you're not averaging across two alleles. Yeah. And what they showed is that as a result of their epigenetic reprogramming program, a lot of what seems to be happening is that you're restoring allele-specific methylation. So you start with a diminished amount of allele-specific methylation. You go through the reprogramming. You have populations of cells that successfully or unsuccessfully reprogram, and in those that successfully reprogram, you're getting an increase in allele-specific methylation versus those that don't reprogram. So the process of aging reversal is kind of a restoration of what you started with. This was my interpretation of that data.
Okay.
So yeah, it's fascinating.
This may or may not be a relevant question. The allele-specific modifications, is there a clear, I realize the cell isn't thinking about it, but what do we know about why it chooses one allele over the other? Is it parental origin or is it always one and not the other? It could be either, but it's one or the other in any cell.
Sure. So obviously, you get alleles from parents, and so you can have two different alleles. That's sort of fundamental. Right. Why there are different levels of methylation associated with different alleles, we get into the realms of speculation. I would expect it to be a function of fundamentally enzyme characteristics, enzyme affinities, if you like. So methylation is deposited by DNA methyltransferases. Hydroxy is a TET enzyme which catalyzes the conversion of MC to HMC. If you have an allele that's different, that changes the binding affinities of those enzymes, then you're going to get a change of epigenetic status. And there's literature that indicates that a lot of allele-specific methylation is within around 60 to 100 bases. So you can kind of conceptually think about that's probably within the kind of space that an enzyme might operate. So that's kind of, I guess, number one. The other piece is that maybe those and this is totally speculative. I don't know what we're talking about here. I'm just going to speculate to the moon about it. But anyway, that maybe those alleles have some kind of impact on the organization of the genome that then impacts accessibility, for example. But we don't know. We don't know. Right. Allele-specific methylation is hard to determine. Certainly, getting it on the same read is pretty novel. So they are good questions that we'd love someone to try and answer.
Yeah. So you mentioned reprogramming a bit ago. Mm-hmm. And I know, so I'm going to move into iPSCs, which I know just enough about to be dangerous.
Okay. All right.
And Tell me if this is relevant or interesting, or do we know anything? So it was my understanding when I learned about iPSCs that to reprogram, for example, skin cells to become iPSCs, there's only a handful of transcription factors that somehow you can add to the medium and reprogram them. Is there anything there related to DNA modifications in terms of MC and HMC?
So that is essentially the reprogramming thing that this customer did, who saw this increase, this restoration of allele-specific methylation. So I think there's surprisingly few transcription factors can have a restorative effect, certainly in my mind.
Yeah.
That restorative effect can I assume manifests across many different levels, and one of those levels is methylation. If the hypothesis is that what you're doing is you're restoring the control of entropy, you're reducing the entropy within a system, because that's what life tries to do, then one of those elements of entropy is going to be methylation. Again, the way I think about methylation, there's different layers of epigenetic control. You can have, some people think about transcription factors themselves as being a layer of epigenetic control. Histone modifications to DNA, and I kind of have in my mind that so I'm going to maybe back up a little bit. So enhancer regions of the genome are known to be marked by different histone modifications and also different methylation statuses depending on how active they are. And in the literature there are kind of four states. You have a repressed enhancer or a repressor, which are highly methylated and also have specific histone modifications associated with them. You then have kind of two in-between states. You have, I think it's a poised enhancer and a primed enhancer. The poised enhancer seems to be associated with different combination of histone modifications, so possibly different to the primed. My histone biology isn't on point, so I don't know. I'm not going to be an authority on that. And then you have an active enhancer, which is demethylated and also has a different histone modification status. It's completely opened up and it's starting to be expressed. With the poised and the primed states, the poised state is a reversible state. So I think it mainly manifests in histone modifications, and it can go one way or the other. Whereas a primed enhancer manifests in a combination of histone modifications and cytosine state modification status. So at a primed enhancer, you start to see an increase in HMC. And because we know that once you've started to see HMC, it can only go in one direction, and that's towards demethylation. As soon as you've started to have HMC sort of deposited by the action of TET, you've kicked off a process that can go one way and one way only, and is effectively irreversible. Certainly for a period of time, it is irreversible. So a primed enhancer is not active, it's about to be active, and it's going in one direction and one direction only. So it's already committed itself to activation. Whereas a poised enhancer, it's not repressed, it's not active, it's in an in-between state, but it can also flip back to being repressed. And what that makes me think is that histone modification statuses are probably more transient. They are more like malleable, if you like, than cytosine modification statuses, because I know, again, I'm out of my depth here, so someone's probably going to tell me I'm wrong, but they're probably more reversible than the methylation status of a cytosine. And
so a little more fine tuning on the histost.
Yeah, potentially, exactly. Yeah. So going back to the kind of why does reprogramming manifest in DNA methylation? I kind of think of DNA methylation as a almost like a record and a slightly more permanent manifestation of epigenetic control. So histone modifications are probably more malleable. Transcription factors, certainly there's malleability there. Whereas DNA modification status is probably more permanent and less transient than some of those histone mods. I mean, HMC is a transient modification of cytosine, but the difference between modified and unmodified is probably more permanent. And that kind of takes me back to, again, some of the Emily Hodges work that I was talking about earlier, where she's looking at the relationship between methylation and accessibility and so on. And what she's shown is that you can not only fingerprint the sort of have a fingerprint to identify a tissue. So you have a different modification fingerprint for cardiovascular cells versus skin cells and all the rest of it. But you can also use unmodified enhancers as a kind of fingerprint of the lineage of a cell. So you have these long-lived unmodified enhancer regions that not only tell you what the tissue type is now, but also tell you where it came from. They tell you the sort of the tree of life, if you like, of that cell type. So again, if we take it back to thinking about cellular reprogramming and restoring, obliterating entropy and restoring order, you're almost going back up that tree, and methylation can almost serve as the kind of the imprint and the record of the youthfulness or the coming back to stem cells, a sort of potentiality of a stem cell. Methylation is kind of the record of that. That's slightly less. That sort of has that sweet spot between the 4-base genome, which is static, the more malleable, certainly more malleable proteome, RNA, more malleable RNA, histones, and so on. And it's got that sort of sweet spot of being changeable over the right sort of time scale to act as a decent kind of record of what should be instantly expressed.
So when you say record, so here's what I'm thinking. You potentially, if I understand what you're saying, you look at a cell and there is a record of its developmental pathway, like a life history.
Yeah.
To say this type of cell came from a cell that looked like that, and a cell that looked like that, and a cell that looked like that, all the way back. I mean, that's fascinating in itself.
It's really cool.
Without having to follow those things as they happen, to be able to look at something and go, oh, we can see, what the origin of this was.
Yeah. And in fact, there are, I think there's so coming back to oncology, there are people who use the TracerX program does this. You have your kind of clonal and branch mutations that allow you to trace lineages within a heterogeneous tumor sample, right? You could think of it similarly epigenetically as well, where you could use an epigenetic fingerprint to trace things back and look at dominant and recessive clones and so on. And then you kind of couple that together with papers that have come out recently that show that you can have cancers that are driven by epigenetics alone. There's a literature from Mark Dawson at the Peter Mac in Melbourne talking about how you can have limited or no genetic clonality within a genome, within a tumor, but where you instead have epigenetic clonality within a tumor, and therefore you can have resistance arising to, where there are no genetic changes, but where you are selecting for an epigenetic clone rather than selecting for a genetic clone. So it all kind of ties into this fingerprinting, this developmental cascade that works really well in embryogenesis and sort of normal development. And then when you get to disease states, like especially cancer, you get this like perturbation, this kind of aberration that is bad development, development gone wrong, development being reversed, and then kind of going into its own developmental tree as you get these different epigenetic and genetic subclones that interact and drive resistance. So, yeah, it's interesting.
Yeah, it's fascinating. Okay, so I'm going to leave you with one last thought. So in the last week or two, I have seen on YouTube and LinkedIn different, let's say, posts or videos, and that have gotten my attention. And I first want to say, oh, that's garbage, because people claiming that like DNA is not the blueprint for life, which I still think it is. But I'm starting to see how the other factors actually, we're starting to understand the importance of all those other things. And yes, at some point it comes down to the DNA, but there's so many more layers on top of that. DNA has to be the final record of truth in a way that you start with, but you can start to see how those changes that you're talking about and other things outside of the DNA are so essential.
Yeah, I think you can almost think about, well, the way I sometimes think about it is like that your 4-base genome is possibility space.
Yeah,
it's the possibility space, but
but not entirely determinative, as we might have thought.
Yeah, exactly.
50 years ago.
Exactly, and it's the interplay between that possibility space and Yeah, between that and the environment, and then the early development, the environment is the embryo. It's driving its own. It's driving its own narrowing down of that possibility space that's manifesting in methylation, and then being driven by a cascade of methylation, if you like. Or in later life, it's being shaped and driven by the environment instead. So yeah, that's how I think about it.
Nice. Tom Charlesworth, this has been absolutely fascinating. Thank you for sharing all of that with us. I'm hanging on by my fingernails on some of this, but it's really interesting and fun to think about. So thanks so much.
It's fun to speculate and get excited about science.
Exactly.